Well, the title of Jason's talk today is How 3D Morphology is Impacting the Lissamphibian Origin Problem, a topic that I'm very interested in. So the floor is yours.
Great, thank you very much. And thank you also to Sarah for seating me this talk. I think she had ulterior motives to avoid too much audience fatigue because she's got another seminar coming up in the near future.
About a month, yeah.
So thank you very much for that. And thank you all for attending. This is meant to be a fairly informal survey of work that we have ongoing in various stages in my lab. And if you have any questions, feel free to jump in and interrupt me at any point. And I'd be happy to answer questions as we go. What I'm going to do basically introduce the nature of what we call the Lissamphibian origins problem. This will involve a little bit of paleontological alphabet soup if you're not familiar with some of the extinct amphibians that I encounter on a daily basis. I'll go over those briefly. Introduce also some relatively recently published new information that informs upon this question, and then taking a look at ongoing research that we have in my laboratory right now using data derived from three-dimensional modeling and CT imaging, looking again at this question of the origin of modern amphibians. Some of these slides are designed for a more general audience. I don't think I need to introduce extant, or what I'll call Lissamphibian, for the duration of this talk. Extant amphibians, this is where I come to borrow them in order to CT scan them. One group you may not be familiar with are the Albinerpetontids. They're an entirely extinct group of somewhat questionable origins and relationships, and we've only missed out on having them in our modern fauna by about 12,000 years. Perhaps a little less familiar to you is the cranial anatomy. I'm going to be focusing primarily on cranial anatomy, although we'll talk a little bit about postcranial anatomy in a bit. Frogs are characterized by having these very broad, open, fairly lightly built skulls. In dorsal view, you can see the orbits are greatly expanded, and in the palates there's a mirroring large opening in the roof of the mouth. So, the fossil record of frogs starts with a single specimen of, again, questionable geologic provenance that was apparently found within a chieftain's hut, not actually found in situ in Madagascar, but it is presumably from the early Triassic, and it is clearly frog-like in a number of features. Again, you have this very broad, openly constructed skull, and you have the fused frontal parietals that are characteristic of frogs. But it lacks a lot of the adaptations related to hopping. It's got an extended vertebral column. It does have these anteriorly inclined ilia, but it lacks a lot of the extension here in the tibulari and fibulari characteristic of hopping frogs. That doesn't arise until the Jurassic. Salamanders have a generally similar construction to the skull, although they're characterized by having an incomplete cheek. The lower dental, the upper dental arcade does not connect up to the back of the skull. But otherwise, again, you have these same large expansions of the orbits, relatively expanded palatal openings as well. Fossil record for salamanders begins later in the Jurassic. Fossils from Asia, Carorus imaged here, as well as Cocartus, which was recently published in detail a year or two ago. But again, it is very primitive in its construction. In the vertebral count, it's still similar to what you see in a lot of the fossil groups, but it does have this lack of connection between the upper jaw and the rear of the skull. So it's an archaic salamander, but clearly salamander. Caecilians, on the other hand, have skulls that are highly modified for their mode of locomotion, which is head-first burrowing. So instead of being broad, expanded, and open, they are narrowed and they are consolidated, characterized by a number of fusion events, numbers of different centers of ossification fuse up fairly early in development, both in the rear of the brain case as well as in the palate and lateral portion of the skull. They're characterized further by having a unique jaw opening mechanism that was described in some detail by Morphology, who unfortunately couldn't make it today, and as well as a unique sensory structure associated with modifications of the adventitia of the eye, the tentacle. The fossil record of caecilians is sparse, to say the least. The first fossil we find is Eocaecilia. It's a relatively complete fossil. We don't find the next caecilian fossil for another 40 million years subsequent. And what's exciting about Eocaecilia is that it preserves a number of plesiomorphies that help polarize characters within later occurring in modern caecilians. So we retain both fore and hind limbs. The skull seems to be much more, much more generalized in its construction. It's showing some of the trends towards the consolidation that you see in later occurring or modern caecilians, but enables us to make some statements about the evolutionary mechanisms that are going on in the origin of caecilians. It's clearly a caecilian, however. It does possess that same unique jaw opening mechanism, the long retroarticular process in the lower jaw that you see here. It has hints of a potential tentacular groove associated with the anterior portion of the orbit as well. And recently published, and I'm not showing any of these slides in this talk, but recent work we've done focusing on brain case morphology show that it actually is clearly caecilian in its internal morphology there. Now to step back, putative potential sister taxa in the fossil record fall out into two generalized groups, and I throw this horrible paraphyletic term here, labyrinthodonts, out because it still persists in the paleontological literature, especially associated with these discussions. Labyrinthodonts was a term that was erected to describe archaic fossil amphibians that had a particular pattern of infolding of the tooth enamel and dentin. They were further categorized by the relative construction of this multipartite vertebra, having a larger or smaller intercentrum versus pluricentrum, whether they were completely crescentic or whether they were still paired, relatively similar in size, led to a number of different ways of categorizing. Generally, labyrinthodonts are large to medium-sized animals of fairly archaic construction. Another group that figure into this question of modern amphibian origins are the Lepus Fondles. And these tend to be small and are united by a single unipartite vertebra fused, typically, although sometimes sutured, neural arch to a complete single-piece centrum, although sometimes in some taxa they'll preserve a remnant of an intercentrum similar to some amniotes today, and a number of other features that suggest that Lepus Fondles are a natural group. Specifically among quote-unquote labyrinthodonts related to the origins of modern amphibians are what are known as amphibamid temnus fondles. So these are very small, actually about frog-like, maybe salamander-like in size, and they have a number of features that weigh into this question. They have large expanded orbits, very large rounded vacuities in the palate, and a number of other features that we'll get into in more detail subsequently, especially focusing here on occipital condyles and the nature of the articulation of the premaxilla to the rest of the skull roof. And in fact, when you arrange amphibamids along with a suite of other more basally occurring or earlier dichotomous branching points within temnus fondles, you can see a general trend towards the reduction in the post-orbital portion of the skull. And you see this, this trend throughout Amphibians, leading towards where phylogenetic analyses would place modern amphibians, or at least frogs and salamanders. This trend, this progressive loss of this post-orbital region, is also seen and is hypothesized to be associated with the origins of frogs and salamanders, at least. But it's still, it's a mighty large jump to get from here to here. There's an awful lot of evolution that's taking place. And it's within that lack of data, which is where this problem of Lissamphibian origins arises. Among the Lepus Fondles that figure into this discussion are these two groups here, the Lissamphibians and the Microsaurs. And depending upon which hypothesis that you're discussing, one or the other of these figures more prominently. The Microsaurs are the ones that are suggested to be related to the origins of Sicilians, and similar to that figure I showed you of Lepus Fondles. It's being argued that you see a progressive consolidation of what is already a fairly consolidated skull towards what was seen in Eosicilia and then on into modern basally located Sicilians such as Ichthyophis. The other Lepus Fondle group that one particular research group has suggested is related to the origins of modern amphibians are the Lissamphibians. These are elongate, reduced limbed, very specialized skulls in this group. But this is the group that has placed its sister taxa to Lissamphibians in this particular hypothesis. So the problem is, what is the nature of, or which archaic group of fossil amphibians is sister taxon, or gave rise to modern amphibians, and how many steps did this process take? The majority view is that modern amphibians are a monophyletic group. They form a good clade, and are sister taxa, if not deeply nested within Lepus Fondles, such as the amphibians, such as Doliserpeton there. There's a minority view, again, one particular research group that argues, well, modern amphibians are monophyletic, yes, but instead their sister taxa are the Lissamphibians, imaged right here. Still another minority view, which takes a number of different forms, suggests, well, it's a little of column A and a little from column B, where frogs and salamanders have origins amongst various groups of Amphibian Lepus Fondles, but Sicilians are sister taxa to Microsaurs, in particular, taxa such as that. The reason that this controversy persists is because we have two non-overlapping data sets. So modern amphibians, most analysis, we can utilize molecules. We have access to a larger suite, even of morphology, with getting into soft tissue that simply don't exist in the fossil record. So in trying to find, these are a number of synapomorphies that were proposed in a seminal paper by Parsons and Williams back in the early 60s, arguing for monophyly of modern amphibians, which up until that point was not generally accepted. And there's a number of good soft tissue-based synapomorphies that are located there, but when you start exploring the fossil record, we have to eliminate all these. We simply don't have access to these lines of evidence, or at least not directly. What we do have, potentially observable from the fossil record, are these two features here, pedicellate teeth and the Operculum Plectrum complex. And that's, well, I'll define that in just a moment. Pedicellate teeth is a particular construction of a tooth, where we have, here, image there is a salamander tooth, and we have a dividing zone, which you can see, here this is a lingual surface that actually is a area of non-ossification, and then a zone of weak ossification that separates the enamel-covered tooth cusp from its base or its pedicel. And we know that this is actually non-ossification, non-mineralization in this area here, rather than an erosional pit for tooth replacement, because we can actually observe that taking place down here. Teeth are otherwise replaced in a fairly standard tetrapod sort of way. The Operculum Plectrum complex is associated with hearing in modern amphibians. So here we have, there's a tympanic ring and the tympanum suspended between it. And here's in yellow, the stapes that then articulates with the tympanum via cartilaginous link. Associated with the footplate of the stapes is a secondary ossification, although it's cartilaginous in salamanders, known as the operculum. Not homologous with the operculum of a fish, obviously, although it takes the same name from the same root word. This is attached to the rest of the skeleton, to the pectoral girdle, via this muscle here, the opercularis, which is a slip off of the levator scapulae. Experimental work has suggested that these two different sound systems break apart high-frequency auditory sounds, calls of frogs, versus low-frequency or environmental sounds that are being transmitted to the inner ear via vibrations picked up through the limbs. So that's the background. A few years ago, I had the privilege of describing a particular fossil that is proving to be quite informative of this question. It actually slits into the gap between some of the Lepus Fondles and a full frog-like condition. This is Garabatrachus haughtonii, which we described in 2008. And the skull itself is quite a bit closer to what you see in modern amphibians. This is an extremely lightly built for a Lepus Fondle. You can see that the palatal vacuities are extremely broad, that this bone here, the pterygoid, has become very lightly constructed. The palatines have been reduced to transversely oriented bars. This is something that you see in modern frogs. The teeth are reduced. Typically, Lepus Fondles have large fangs, the labyrinthine enfolding, although that's lost within the smaller Amphibians. But here in the premaxilla, this bone right here has 30 separate teeth. It's very unique amongst Amphibians. It also has a number of other unique synapomorphisms otherwise seen only in modern amphibians. Firstly, it has, I've argued, pedicellate teeth, and you can tell that based upon the presence, and I think it's fairly clear to see here, of a dividing zone. Now, this observation has been criticized in the literature. One criticism is, well, the figure doesn't really show it particularly well, and it's hard to shoot at this high of a magnification. At this point, my microscope is picking up vibrations of the refrigerators across the hallway. I just can't inertially dampen it enough to get a nice clear picture. But the other criticism that's, I think, a little more valid, and one of the reasons why I'm here for my sabbatical, is related to the nature of the construction of the pedicel here. And it's been saying, well, that's not quite right from what you see in modern amphibians. That's generally, they're parallel-sided, they tend to be taller, these are kind of short, kind of squat, and it's probably not it. I think there's a lot more variation in modern amphibians, and speaking with David and Marv Lee, they agree. So we're doing a survey of morphology of pedicel to cusp morphologies in modern amphibians to address this particular criticism. Also, one of the neatest things about this fossil is the hind limb. Now, this fossil is fairly well articulated, although not completely preserved because of differential erosion before the fossil was found. The digital count is exactly what you see in both the Amphibian and Lepus Fondles, as well as the first frogs and salamanders. So Tridobatrachus, Carurus, Cocarctus all have the same digital phalangeal count. But it also preserves a couple of the distal tarsal elements. And this is exciting because of where they're located and what the probable identity of these specimens are. So we have one, you can see right here, large element that articulates with the base of the first two digits, and then at the base of the third digit here, there is another, and in fact it actually shows not as strong of ossification. It's not completely ossified. We've identified this as the effused distal carpal 1 or 2, which is known as the Basale communa in salamanders, and as well as a distal tarsal 3. This we excavated away from the matrix. It was a large piece of matrix. We cleared it away. There are no other elements associated with this ankle. Why is this important? Well, this is a unique synapomorphy otherwise only seen in salamanders, and it is suggestive of a particular developmental mechanism of digit development. Most tetrapods, frogs, amniotes, develop their limbs in a stereotyped way, where proximal elements of the limbs, so humerus, femur, and then ulna, radius, or tibia, fibula, ossify first, and then the proximal carpal and tarsal rows, and then the establishment of digit 4. And digit 4 that then leads to digit 3, digit 2, digit 5 will come in around that point, and then digit 1. So establishing a digital arc at digit 4. And proximal carpals and tarsals first. Salamanders, on the other hand, again, these proximal part of the limb appear typically, but then it skips to the distal tarsal 1 and 2, the Basale communa. This establishes the digital arc. This is a so-called pre-axial pattern of development. Digits then are acquired in an anterior to posterior direction. So digits 3 and 4 and 5 will come in, digit 1 and 2 being established there, Basale communa. Additionally, it is these distal tarsals that also ossify first. So Basale communa, distal tarsal or carpal 3, and then 4, and then subsequently you get the proximal elements. So this is exciting because this is suggestive of a very clear synapomorphy with modern amphibians. Now, this observation has again been criticized. Perhaps we've misidentified it. Maybe it actually belongs to this element Y here, but that would then require an entirely new. It would require one of two different assumptions. One, that something would leave this element in place, but then remove elements that would typically appear first. And given the fact that the rest of the specimen is so well articulated, we think that's unlikely. The other assumption would be, well, distal tarsal Y is appearing first before all the other carpals or tarsals in this case, which would invoke a pattern of digital development that is otherwise unknown in tetrapods. So we put this specimen into a data matrix that I had assembled back in 2001, and we added in modern amphibians as well, and I'm just going to focus in on these two boxed areas here. Our results from this paper in 2008 suggest Garabatragus is sister taxa to batrachians, so frogs and salamanders exclusive of Sicilians, and Sicilians themselves are displaced within these microsaurs, within these Lepus Fondles, specifically associated with two particular types of microsaur, which is an unanticipated result. It is supportive of this polyphyly hypothesis. So little from column A, little from column B. But as was immediately pointed out again in the literature, this flies in the face of molecular phylogeny, which strongly support modern amphibian monophyly. And were this suggestion of polyphyly to be correct, you would expect Sicilians to be displaced closer to amniotes than to frogs and salamanders, and you just don't see that. Our feeling is, well, yes, this is a problem and we need to explore it further, but just rejecting it out of hand based upon that ignores the fact that there are statistical anomalies related to rapid divergence events followed by long branches. In this case, the long branches would be on the order of 300 million years. So we felt that this merited additional exploration. So we're looking now in more detail at trying to find new anatomical systems and other lines of evidence that we can bring to bear in this question. And to do this, we are using new imaging modality using X-rays. So we're looking closely on the inside of what are going on within these fossil amphibians, and what we're finding is actually relatively surprising. So, first thing that we did, started looking at these various groups of microsaurs. One of the problems we have with fossils is that they tend to not be very common, and to study internal anatomy, especially brain case anatomy, which has recently been shown to be relatively resistant to a lot of the selective pressures that the rest of the skull will undergo related to locomotion, related to feeding, etc. So we wanted to start characterizing what's going on in brain case anatomy, but traditionally getting access to those data required the destruction of specimens, and museums tend to be reluctant to let you do that when they only have a couple of a particular taxon. Especially in the case of Garabatrachus, that's the only known specimen in the world, and they're not going to let you serial section it just to see what's going on inside the brain case. But CT, modern CT, desktop CT is now relatively affordable, and I hate to say $500,000 is relatively affordable, but it's the sort of thing that is within the reach of research groups now. So we've been borrowing these fossils and we've been CT scanning them, and we're finding an awful lot of really neat information. So this is one of those microsaur groups, and you can see it's got this overturned snout. I've named this general clade Recumberostra for this. And it has this unique, you can see a portion of the brain case sticking out here. Unfortunately, the rear part of the brain case is kind of munged up. But the front part here is actually pretty neat. And from the original description, we couldn't tell whether this is a flange coming down here off the frontal bone that just overlapped the brain case, or whether it was a separate element. So we CT scanned it. Let's see if I can make these embeds work. So you have to have a spinny animation when you do CT work. It's just required. It's like if you publish on a dinosaur, you have to compare it to T-Rex, right? You have to have a spinny element. So you can now digitally dissect away the parts that you can see in the hand sample to get to the parts that you can't see. So this is the anterior portion of the brain case here, and it turns out that this element is a separate ossification that then itself would articulate down with the frontal. So here we have the sphenoethmoid, and we have this questionable element of questionable homology here, parasphenoid line here. And it's got an interesting pattern of overlap with the sphenoethmoid that suggests that it's relatively resistant to torsion and compression. And if anybody's interested in doing a PhD on biomechanics of microsaurs, please see me after the talk. But as a single example, so we started expanding. We have to get a better sense for the diversity of brain case morphology in these animals. So we digitally dissected away. This was sister taxa to Sicilians in my phylogeny. And again, you see this multipartite sphenoethmoid and this questionable element here associated with the rear portion of the brain case. And the construction of the brain case is very distinct and very different than what you see in modern amphibians. And we expanded it additionally. Here's a gymnoarthroid cardiocephalus. We dissected it away. You can see that we retained the same pattern of overlap, but this time it is actually a lapid of the frontal, and it is just sphenoethmoid. So there's no additional ossification here. So in gymnoarthroids, and this is another taxon that will be out in the next JVP, which is a basal, also I believe is a basal to the first one that I showed you. Again, lacks that ossification. So it suggests that ossification is acquired multiply within microsaurs themselves. Here again is corolla and the first one I showed you, nanorotor, where they have the additional ossification, but they're acquired differently. The nature of the articulation with the frontal is similar to what you see here. So it's this part of the brain case goes inside there. So we're getting a better sense for the diversity of brain cases within microsaurs. And in looking at the nature of the characters that are linking Sicilians with microsaurs, we discovered none of these brain case characters are involved. It's all related to external morphology associated with all animals that burrow head first. So this is convergence. You see it in amphibians, you see it in skinks, you see it in burrowing snakes, you see it in these various microsaurs. So the nature of characters that were pulling them over seem to be related to convergence. Now Hilary Madden spent some months here a couple of years ago working through both CT scanning a number of Marvelese Sicilians, as well as working the way through the histological sections to see what structures pass through these various holes. And she characterized very clearly the morphology of the rear of the brain case and the anterior brain case. Very different sort of construction. It's a different nature of articulation here at the top of the anterior brain case with the roof of the skull. It's completely different than what you're seeing in the microsource. Neat thing that she discovered, though, is when looking at the Sicilians themselves, that you see variation in the sorts of openings here in the same area of the lateral portion of the brain case. And going through and comparing these patterns, she was able to cluster into 8 discrete morphologies that actually map beautifully onto molecular phylogenies of Sicilians, as opposed to some of the morphological phylogenies. The neatest part of her work is that she established also a developmental mechanism for acquiring these different features. So here is a single growth series of, I think this is Dermophis, that she reconstructed from histological slides in 3D, and it shows progressive acquisition of these various morphologies at various developmental stages. So a particular event of heterochrony can arrest, if it arrests this particular region of the skull, can lead to these different, otherwise phylogenetically informative morphologies. It's a pretty neat piece of work. But while doing that work, she also, with a 3-dimensional representation of the skull, she realized that you can create endocasts of the inner ear, and that there may be an interesting morphology associated with that. And keep in mind the labyrinth system has a number of different epithelia that are sensitive to various frequencies and sensations. And so going through creating endocasts of the endolymphatic capsule within Sicilians, she noticed a few features. One, that Sicilians have a number of strong synapomorphies with other modern amphibians that were previously unappreciated. And another hypothesis, for instance, of the medial pressure relief system had been proposed to have been passing through one of those holes in the skull that she had described earlier, and she in histology showed that it's just passing various nerves and blood vessels and not relieving the inner ear. So it actually, strong synapomorphies, again reinforcing monophyly modern amphibians. But she also noticed that there was progressive loss of 2 of these sensory epithelia that are associated with the lagina and the basilar papilla. And these are associated with the opercularis, or low frequency, and high frequency sound system within frogs and salamanders. Which means that at some point in the evolutionary history of Sicilians, they must have had these hearing systems in order to lose them through the clade. So when she mapped both of the synapomorphies as well as the observations of the acquisition and loss of the sensory epithelia onto these various hypotheses of modern amphibians, the only parsimonious interpretation is that there's a single evolutionary origin. It's the monophyletic origin of modern amphibians. But of all of these groups, you only see the Lissamphibian ear in these Temnus Fondles. So when she actually takes these observations and plugs it into that 2008 matrix that found support for polyphyly, those were sufficient to reestablish modern amphibian monophyly within Temnus Fondles. So, convergence drives, we have to be careful when we're constructing our phylogenetic analyses, because convergence is real, may be unappreciated until you start digging deeper into. So don't take a cladogram spit out at you by Palp as sort of the result. It's the beginning. It's the hypothesis that you're testing. It's important to remember these are just hypotheses. It's not. We hope it approximates reality, but it is not reality. Really briefly, we've got a lot of other ongoing work. We're trying to characterize what's going on in salamanders better. We are creating a 3-dimensional morphometric analysis of growth in a single species of salamander. This is Embystoma macrodactylum. We actually collected the eggs, raised them in the lab so that everything is controlled, because in these salamanders, rates of growth vary from locality to locality. So, was talking with Jesse earlier, the issue of correlation with size breaks down often when you're dealing with developmental data. So we're working with CT. These are synchrotron data, but you can actually see the snout is somewhat compressed here. It's an artifact of the centrifuge tubes. So the synchrotron data are basically not usable for the 3D morphometric analysis, unfortunately. So I have a student who's right now busily setting landmarks all over these skulls, and I hope to have some more results to show you here, but this is a long process. It takes a while to ensure that your landmarks are homologous. But once we have this generalized sort of salamander in this one species, characterize the three-dimensional shape change through growth using these staged embryos, these staged larvae, then hopefully we can make comparisons with other salamanders of different life history, different developmental modes, and we can actually narrow in on what shape change is taking place in these various different life histories. So, you know, ongoing data. We're also able to take fossils and we're able to digitally dissect out missing soft tissue data. So you can see there's a series of holes here that are leading to branches of alveolar nerves and blood vessels and the like. So you go through in your computer program and you can fill in these holes, and that actually provides interesting data as well. And one of the things that this is early days in this research, one of the things that we're discovering is amongst these microsaurs is that there is a trend within this clade of Recumberostrans towards more and more amniote-like pattern of tooth implantation and replacement. Whereas amongst these Lepus Fondles, there's a progressive trend towards more and more Lissamphibian style tooth implantation development, acquisition of that pleurodonty, and ultimately into taxa such as Doliserpiton here. Unfortunately, Gyropetrachus doesn't CT scan very well. It's got too much iron in the matrix. But Doliserpiton has tooth implantation otherwise indistinguishable from modern amphibians. They are pedicellate, they are bicuspid, they meet all of Tron's criteria too, of the pedicel being parallel-sided, etc. We're also looking at Lepus Fondles, and these are just a couple spinny animations as I come to a close here. This is Brachydactylus. This is the Lysarophian that is purported to be sister taxon to modern amphibians, and we thought, what better candidate for exploration than this? Here's a 3D spinny animation that was produced less than a week ago. So this is hot off the presses. Brain-cased construction. It's a microsaur. It is very similar specifically to the Brachystelechids, one of the ones that was found to be proximal to where I had originally, 2008, suggested Sicilians belonged. The brain case morphology is dead on to what you see for them. And there's other features associated. You can see how thick the skull roof is here. That's real, and it is also present within these Brachystelechids. So this is a derived Brachystelechiid microsaur, but derived for what? And we will suggest, and this isn't even that terribly radical, because you find these Lysarophians in paleosols in estivation burrows. Okay? So they're soft substrate burrowers, and if you note here, you see the same morphologic, at least in terms of external anatomy, in modern amphibians that burrow in soft substrate. So this is Amphiuma. This is Tridactylum, I think. They burrow, they estivate. So there's, a superficial similarity all the way down to the massively ossified hyoid apparatus, which are not associated with lungs in either of these taxa. In fact, Amphiuma, I think, lacks a tongue as well. So this, again, it's early stages, but we seem to have found external morphological correlates to why exactly this morphology is being derived. So again, convergence due to shared locomotor habits. And that is, that in the loss of bone elements between these and in those gaps between archaic fossils and modern amphibians is what's allowing the modern amphibians to be placed with these in that particular data set. I'm actually in the process, one of the things I'm doing here is my sabbaticals, and again, I'm CT scanning as many frog salamanders and Sicilians as I can get a hold of, because I'm hoping to add the morphological data to the molecular data as well, to create a total evidence analysis. But these sorts of observations are a side product of this work. Finally, and this is again just hot off the presses. This is kind of neat. This is the Istopod lithyscus, earliest known tetrapod before the East Greenland fossils, Acanthostega and Ichthyostega, were found way back when. Now we know a number of other ones. It's found in an ironstone. In 2001, I guess I published my first attempt at analyzing CT data. Our tools have advanced tremendously since then, so we decided to take another kick at these data. And what we found is that it is an extremely primitive skull. I mean, it's very derived, very lightly built. We've reduced a number of the ossifications that are otherwise present. But the lower jaw morphology, we can now code this, and we dropped it into Peralberg and Jenny Clack's matrix of early tetrapods. It falls out at the base of the tree. It falls out down towards Bifidids, Wachiriids. And when we dissect away the dermal elements of the skull, so next slide, I'll give you a little spinning of the brain case. It's fascinating. We have a notochordal pit. We have a maintained opening of the intracranial fissure. These are primitive characteristics. We have this huge dorsal lappet off of the parasphenoid, restricting the brain case to the top. You see these in Bolomeres, you see these in Wachiriids. You don't see these in derived microsaurs. This suggests very strongly to us, and again, these are early days in these analyses, that Lepus Fondles themselves are not a natural group, and that we have convergence being driven by small size, similar habitats, potentially, and an over-reliance on the postcranial characters that are remnants of sort of pre-cladistic systematics. So with that, thank you very much. I really appreciate your attention. I just wanted to emphasize that in order to get at these problems, I mean, we can spend a lot of time analyzing statistically one matrix versus another, but it's observations. It's observations of fossils, it's observations of animals in jars. It's looking at things in new ways, finding new lines of evidence. It's these observations that are so key to breaking down these sorts of questions. Thank you.
What becomes of microsaurs in your latest analysis, and then what happens if you take those homoplastic characters that you identified and re-ornament the tree with respect to those characters? Do you learn anything about ecology?
Again, we're in early days. The only microsaurs that I'm confident to say are, a natural group are these Racumba rostrums. I'm starting in a narrow group where we can actually make good observations and expanding out from there. So things like Microbrachus, things like Utah urbiton, which are, they're not very, they're derived in other sorts of ways. And I wouldn't be surprised. I mean, and even my analysis, Marcello's analysis, Marcello Ruta and colleagues have done a number of detailed phylogenetic analyses. And we're finding that Lepus Fondles, they bounce around an awful lot. And it's those particular taxa as well that aren't well pinned down. And I don't have a lot of confidence that a lot of what we would call microbrachomorphs belong with a lot of things like gymnarthrids and pantylids and the like. I'm pretty sure that those belong together. But again, they all share these same morphologies associated with head-first burrowing of some degree or another. So there's a lot of work that has to be done with respect to Lepus Fondles in general. Now, my graduate student Jason Pardo has given presentations at SVP arguing fairly convincingly that some nectridians are probably brachiopoid or plagiosaur-type temnospondyls instead. And if there are any Diplocaulus skulls downstairs, I can actually show you remnants of a notic notch. I actually think he's onto something there. There are a number of very good features that suggest that they belong elsewhere. Now, so I think that we're due a reconceptualization of exactly what morphologies are present in these Lepus Fondles and how they associate with one another. one thing I will say about Rinconcos, Bob Carroll has argued very strenuously, it's sister taxa to modern Sicilians. It's a composite of two different genera, and not just two different genera, from two separate families. One of the type series, we think, and these are preliminary data that still have to be rigorously explored, but our working hypothesis is we've got one skull that is a brachiostelegid and one that is a basal gymnarthry. So we're getting a better sense for what's going on early in these groups, but beyond just recumber rostrins, I think we need to do a lot of reanalysis. Yeah.
So is it your sense that pedomorphosis may be playing a role in creating confusion with some of the morphological character systems and reconstructing this history?
I think that, so we get into a huge issue here. Pedomorphosis, I think, is what is driving the acquisition of, certainly with frogs and salamander, morphology from amongst Temnus fondles, because you see juvenile, more basal Dicerophoids have traits that are retained in the adults, or at least what we presume are the adults from various other direct or indirect analyses. Skeletal chronology right now is being used by Sophie Sanchez and her colleagues to try to nail down exactly when metamorphosis is taking place in a lot of these taxa. So there's a lot of really neat work that's going on there. So pedomorphosis in particular is a very important for driving the evolution of the trachea condition. But I think what is the bigger confounding factor, rather than pedomorphosis, it is adaptation. And I think it's in particular, you see this with the head first burrowing, and that places extreme constraints upon the morphology, because you now have to streamline. In the case of Sicilians, you need to resist propulsive forces that are, I forget what O'Reilly had estimated, by 200 or so times what most tetrapods would be able to produce in terms of that hydrostatic head first propulsion system. So I think that is the major confounding factor. And in fact, I think that this is one of the strong lessons that come from John Wiens et al.'s paper in 2005, which set out to suggest that ontogeny discombobulates phylogeny. It's the title of the paper. It's a really good paper. But in his analysis, what was causing the biggest trouble wasn't pedomorphosis. It was adaptation towards aquatic lifestyle. So I think adaptation is the major confounding factor. But again, I'm open to pedomorphosis being important, but I also am very sensitive to not trying to introduce my preconceptions of what pedomorphosis is supposed to look like on the fossils. So I try to work with the analysis to produce new hypotheses to go back and look at. Does that answer your question?
Yeah, I guess when I think about it, I think of adaptation to aquatic environments in modern amphibians as generally being allowed by this axis of pedomorphosis. I mean, so those things are correlated in a very strong way.
Mm-hmm.
I was curious about the side-by-side illustration you showed of the Amphiuma together with the Microsaur, mm-hmm. and you were talking about the Amphiuma being a burrower in soft soil. So I mean, it is pedomorphic as well, right? So
Mm-hmm.
like the lack of a tongue and the enlarged hyoid apparatus is probably a bit of a suction feeding in these guys, right?
Mm-hmm.
And so maybe that was the case also with
Absolutely,
Microsaur?
absolutely. Yeah. In fact, they also retain the same elaborate pattern of pharyngeal arches as well. The thing about microsaurs is that in this morphology in particular, pedomorphosis is reflected in the fact that the cheek, the loss of the cheek bones is something that's a late occurrence within microsaur ontogeny. So in some taxa, like the ones I was showing you, the first one we CT scanned, it belongs to a group called Ostotolipids, and they're characterized by having this arc in the lower portion of the temporal region of the skull. And we now have some developmental data that suggests it's because it's either pedomorphosis or they are juveniles, because that seems to be the closure of that is a late occurrence. But additional to that, I mean, this extreme anterior inclination of the lower jaw, that's specialized. I mean, with respect to microsaurs. Yeah, having both the open cheek and an extended skull and there's adaptation that's going on there as well. Great.
Further questions for Jason? That's it? Let's thank him again for the interesting talk.